So, what is gene editing? We asked Ian Affleck, Vice President of Plant Biotechnology at CropLife Canada.
So to talk about how gene editing works, it's important to reflect on how DNA functions in general. So DNA is like a long book of information that instructs how a plant grows. And scientists can now identify the exact page in that book, the exact paragraph, even the exact word they want to change. Then they can use gene editing tools to make that very specific improvement to the plant. So if you were to compare this to conventional breeding, and I'm oversimplifying, but if you were to take 2 pages of a book and you know you want one letter from this page to move on to the other page, but you want the other page to say entirely the same other than that one letter, conventional breeding is kind of like rubbing the 2 pages together until that word, that one and only one letter transfers. Now it's much more complicated than that, but that's essentially what it takes. And it takes plant breeders years and years and years and thousands of plant crosses to achieve that outcome that they know from Mendel's work is possible. They know they can do it. They just got to get it to happen. So gene editing allows them to go in and make that tweak very specifically to the gene that they know they're trying to influence. So a very effective tool. Now, and it's not only useful for plant breeding, it's also useful in human health, industrial applications, and animal health. But today we're really going to focus on the plant side of it. So when we say, so what is gene editing? We're talking about it goes in and makes specific changes, but what really is it? So I really enjoyed this graphic from a recent meeting. The government of Japan used this as they described gene editing during a recent OECD meeting, an international forum on the science of plant breeding and food. So it's a technology that allows scientists to make precise targeted changes to the DNA. And as the Japanese government has put forward, this smiley little CRISPR-Cas9, where we see all the colors lining up, that's because the Cas9, the CRISPR, the gene editing tool is designed to match exactly with the DNA that you're looking for, and then make the cut at the specific area that you're looking to make in order to affect the change that you would have been doing with your conventional breeding activity anyway. So the idea here is to be very focused, very specific, make the cut you need, and as the DNA repairs itself, it'll repair itself in the approach that you're looking for. So a really useful tool in plant breeding to get to those changes in a much more efficient manner. It's important to note two kind of revolutionaries in the space. This is Emmanuel Charpentier and Jennifer Doudna, the two scientists who discovered CRISPR. They saw that this was happening in nature and brought it out to use it as a plant breeding tool. And for any trivia night folks who want to know what CRISPR stands for, it's clustered regularly interspaced short palindromic repeats. I'm sure that will not stick in your head immediately after this, but maybe on some trivia night somewhere that might come in handy. But they're really trailblazers in their field and brought forward a Nobel Prize winning scientific method that's going to have great opportunities in plant agriculture and, as I mentioned, in other fields as well.